Hello and welcome to this webinar. Thanks for joining for your lunchtime. I'm Cara Brosnahan, the Principal Scientist of Animal Health Research, and I'm going to take you through the results from the last 2 years of the facial eczema sheep poo study. After that, we'll have time for questions, and we've got Ginny Dodunski here, a self-confessed sheep geek and large animal vet, as well as the Wormwise Program Manager. We also have Sonia Shaw, who is another vet and a member of the sheep poo study team, who will be taking questions throughout the webinar. So as usual, a little bit of housekeeping. Turning your camera on if your internet is good enough to handle that. So the green triangle there is the button that you click, so that video camera, click to have your camera on or off. So suit yourself on that one. But if you can, mute yourself when you're not talking. So that is that little microphone symbol in the yellow circle. So mute if you are not speaking so we don't get that background noise. Throughout the presentation, if you have any questions come up, you can put them in the Q&A. So we've got these 3 dots here that if you click on that, there should be a Q&A thing come up, or it might be along the top of your screen. So pop your questions in there and then other people can upvote those as well. When we've finished the presentation and we're into the discussion, also if you'd like to just ask your question, you can use these icons here and just put your hand up and we'll come to you. Right. So first of all, massive thank you to the farmers participating in this study. We literally couldn't do it without you and really appreciate all the time that's taken to do this. So facial eczema is only a North Island problem, isn't it? Northland doesn't have a problem with FE anymore. These are really common things that I hear, but what we've found is that they're not actually true. So what we've learned over the last couple of years with the sheep poo study is that we've found facial eczema spores in Otago and Southland right up to Northland. So pretty much wherever we've looked, they're there. The facial eczema risk was still quite high into late May, early June. And we've seen clinical facial eczema in the Marlborough Tasman region north. So again, in each region that people are participating in this study, that's where we've seen clinical facial eczema. And how we've done this is through the sheep poo study. So this is part of a wider program, the Eliminating Facial Eczema Impacts program, where we are aiming to get tools and solutions for farmers to be able to combat this nasty disease. So the sheep poo study itself has got 3 main aims. One is to understand the facial eczema risk, and we're doing that by looking at the spores that come out of the animal in the sheep poo. So that is the risk of FE, doesn't mean that FE is actually there. But we're also looking into the prevalence of FE. So we're asking farmers who are in this study if they've seen clinical facial eczema. We're also going out and doing some testing on farms that have elevated spore counts to see if there is some of those hidden signs of FE or the subclinical FE. And we're using all of this data to build a predictive risk model. So the aim of this tool is to be able to provide better advice in terms of, is this going to be a bad FE season for me and how do I need to prepare? So Awanui Labs are doing all of this testing of the spore counts. So they're receiving a lot of poo from October to May. And in Genome, a company in Dunedin, are building the predictive risk model and also doing the analysis from the data that's coming out of the sheep poo study. We have farmers participating in this research, as I said. So they collect samples from one mob of sheep from October to May every fortnight. And in year 1, we had 203 farmers, and in year 2, we had 278 farmers. And our collectors range in age, as you see. Here's a couple of them from our older participants. So here's Paul in the Wairarapa, through to, this is one of our youngest collectors from the King Country, Oscar. And there's a number of reasons why farmers are getting involved in this study. Partly it's because they find it's valuable research and want to do their part. But also there are people who want to understand better how facial eczema affects their farm and their animals. There's also an aspect of learning how to monitor it better and understand if people do have FE genetics, what kind of challenge their animals might be facing. And if you are a sheep farmer, you can still sign up to the final season. So we still want your sheep poo. You can go onto the Beef and Lamb New Zealand website and look in the Knowledge Hub, search facial eczema or sheep poo, and it should come to a page where you can register. Or there is a QR code here that you can hold your the camera of your smartphone up to now, and it will take you to the link to sign up. So that's the sales pitch done. I'll probably come back to that later though. So going into the results in a little bit more detail. So season 2 results, this is from October 2024 through to May 2025. So just finished. And this is how the spore counts looked across the country. These maps show the highest number of spores that were found in that region that month. Green means there were no spores observed. And then from yellow through to black is increasing number of spores found in the fecal samples. So of note here is that there were sporadic cases of spores seen from October, November, December in the North Island, which is pretty early, but they were low counts. January through to May is where the counts became more consistent. And as you see, this season we found counts in Otago from February to May. Again, at pretty low levels, but still there. Also of interest is that moderate to high risk of spores, so the red maroony colour through to black in the middle of the country through into late May. And here is the map when all of those separate ones from October to May are combined. And when we asked farmers to report their clinical FE, there was approximately 28% of farmers within the study that reported clinical FE this season. And the most was in the Tasman region, with 53% of the farmers there reporting clinical FE. In these other regions, between 30 and 40% of the farmers had FE, and then the lowest reports in Northland, Auckland, and Wellington. So definitely still cases of clinical FE in Northland and definitely in the top of the South Island. We also get the farmers to fill in a survey at the end of the season so that we can understand what other challenges they might be facing and how they are dealing with FE if it is a problem in their area. So this thought might be of interest to people, that internal parasites was the top other challenge that they were dealing with, which is probably not surprising. So this was a combination of, it might be something to do with drench resistance, but it also might be a burden of something like barber's pole. So there was a variety of things there in internal parasites. Pests was the next highest topic, I guess, with purina, and that included purina and grub. There's a lot of different ways farmers were managing FE. So again, this is just farmers within the sheep poo study. And we had 196 of those 278 farmers provide information on this. And of those, 59 were using preventative management. And zinc was the most common preventative management used, either as a bolus or drench. There were other means of providing zinc as well, in feed, in water, lick blocks, and a small portion of farmers using zinc to spray on the pasture. FE tolerant genetics was the next highest tool people were using. And if you are looking for rams with FE tolerance, NProve is a great tool to be able to find them. So if you want to learn more about that, again, you can look on the Beef and Lamb New Zealand Knowledge Hub to find that. And then there were some other things that people were doing in terms of grazing. So, maybe not grazing too low in the sward, using safer areas of the farm where they know there's not hotspots, spraying fungicides, using crops that are safer for facial eczema, and also some using lime. So a variety of ways people are managing facial eczema. In season 2, we also did a further in-depth study, and we purposely did it in season 2 rather than season 1 because we wanted to know what the risk of those potential farms were going to be for this study. So we looked at the data from season 1 and then selected people for this season 2 study. And the purpose of this was to get more data to build into that FE risk prediction model, to get better accuracy so we can provide better advice for these different monitoring tools. So these animals every fortnight were, their blood was taken, feces was taken, and a pasture sample was taken so that we could understand if there were any changes to the liver and redo those fecal spore counts and also look at the pasture spore counts. So this provides information to how related these different test methods are. And we were also using this study to better describe the disease and make sure that if there is an animal with photosensitivity, it really is due to facial eczema and not something else. So we had 32 farms in this study, and this was the distribution of them around the country. And we're still working through this data, but preliminary results show that there was about 60% of them that had possible subclinical FE. And as I say, we're just looking into this to make sure that is correct and it's not due to something else. And then 1% had clinical FE. So we had a range from nothing to clinical FE, which is nice. We had a lot of different breeds in this study. So of the 32 farms, Romney was the dominant breed, but closely followed by Wiltshires, and then lower numbers of these other breeds. So there was certainly a range there as well. Right, so that's season 2 results. So now we'll go back in time to season 1. So this was 2023 to 2024. And this was what it looked like then. So again, the green means there were no spores observed, through to yellow was low spores, and black the highest spores counts. So of note here, we saw nothing from October to December, and from January onwards is when we saw that consistent spores coming through. And yeah, of note here that we found spores in Southland in April, and also saw them in Otago in February, April, and May. Again, that moderate risk was still there into May, so important to think about that when you're thinking about your FE management. I did just want to make a note about the West Coast. So you'll see that on season 1 and 2, they are green, so no spores were observed. We have a very small number of farms in that area, so this is not as accurate as we want it to be. And we have heard of clinical FE cases this season from the West Coast, so just a note there. So this is what season 1 looked like as a combined map. There was not as much clinical FE reported from season 1. Approximately 12% of the farms in the study reported it. And most cases were in Taranaki, King Country, and Bay of Plenty, and then smaller cases around the other regions. But again, still seeing it from Tasman, Marlborough, north, and in every region where farmers were participating in this study. So if we have a look at the 2 years side by side, this was 2023-24 and 24-25. And yeah, I guess the most interesting thing when you see them side by side is that different distribution. So in 23-24, it looks like the highest spores were from that east side, and then 24-25 from the west side, which is really interesting, and we'll do further analysis on that, and it'll be really good to see that 3rd year. And this is why we're doing this study over at least a 3-year period, because of this variability with facial eczema year to year. So for season 1, we also looked into different things that could be a higher risk of getting FE or not. And no single one of these things that I'm about to talk about explains the large proportion of variation in spore counts. Each of them contribute, but it's really important to know that not one thing is going to mean that you have the high risk of FE. It's going to be a combination of things. And this is analysis just from season 1. we'll be using the data from season 2 and 3 to understand this better and confirm these findings. So the first one, we asked farmers every time they submit their samples to say what height their pasture is. So using this data, we saw that taller pasture meant there were less spore counts in the feces. And this makes sense because the biology of the fungus is that it sits in that lower level of the sward, in that dead leaf litter material. And so if your animals are not grazing down into that material, then they are less likely to be able to pick up those spores. We also asked about pasture type, and when farmers reported that they had hill country pasture or native pasture, that was where more spores were seen in the feces. And again, remembering that not one of these things is going to be mean that you have that higher risk, but this fits with what we know about the biology of the fungus as well, because again, on those old established pastures, there's likely to be more litter and likely to be more spores. Potentially they are grazed a little bit harder as well. So all of those things combined mean that there might be a higher FE risk. Looking at elevation, we saw that correlation between higher elevation and less spores in the feces. And again, this fits nicely with the biology of the fungus. If you're up higher, it's going to be a bit cooler. The fungus wants warm, moist conditions. So again, that's just not getting it at those higher elevations. Aspect wasn't seen as significant for the fecal spore counts. So work done in the 60s showed that if you're looking at pasture spore counts, those shady south-facing slopes or slopes facing prevailing drying winds are safer, and that is likely still to be true. It's just we're using a different method, so we're using the fecal spore count, so that animal might be grazing on multiple different aspects. So when we looked at that compared to aspect, there was no significance there. And in terms of looking at your neighbour's property or the regional facial eczema counts that are happening, FE risk was found to be clustered. So if your neighbour had a high or low risk, there's likely chance that you have a similar risk. But really important that your risk won't be the same because of all these other parameters that are needed to make facial eczema happen. So you can use the information from nearby farms to help you guide your own monitoring and decision making on your farm. So you should still check your own counts, and whether it's pasture or feces, especially if you've got slightly different topography or terrain than your neighbours. And here are some things that we heard from the farmers in the study that were kind of interest to them. So just knowing that those spores were so high, so late in the season is really important. And also understanding if they are the toxic spore or not. So that was one consideration. That the fecal spore counts didn't line up with local pasture counts. And yeah, for some of the reasons I just described before, that does make sense. There's so much variability within the paddock, between paddocks, between farms, and your animal is going to be selectively grazing in different parts. And your pasture sample, you're trying to take a representative sample, but we know there's a lot of variability. So we wouldn't expect them to necessarily match, especially if it was your fecal spore count with a regional pasture spore count. And again, that's with the in-depth study. We're trying to learn more about that. So again, we can provide that advice on when and where to use these different monitoring tools. Other feedback was that people quite liked the fact that they took time to check in on their animal health while they're waiting for them to poo. Generally, we ask people to corner their animals and wait for 10 minutes or so they can collect some fresh poo. So in that time, taking the opportunity to actually see what their animals are looking like. And again, some people had some ways of efficiently collecting that poo. So going out fairly early in the morning, and when the mob is resting, and then as soon as they stand up, they're going to poo, and you can collect that fresh feces. And then also someone's put in there that timing is really important. So sometimes you go and they're going to poo, and if not, come back later. So again, the sales pitch, so you can still join up to the final season. We're really keen to get a few more farmers this season, so we want about 300. So yes, please sign up if you're keen. But if you're not a sheep farmer and you have other livestock, but you still want to contribute to this data, would be really keen to hear from you about if you've had FE on your farm this season. Here is another QR code and a link that you can go to fill in a very simple form telling us what species you farm, what you saw in the animals. So that would be really appreciative. And if you know of anyone else who might like to fill that form in, please hand that on. So finally, just a few things to consider. Is your farm at risk of FE? So maybe if you are in an area that you didn't think FE occurred, might be worth asking some questions and just looking into that. Are you covered by your facial eczema management plan? So given that spores we've seen consistently over the last 2 years were quite high late in May, would that cover you if you gave a bolus early on? And we've found that clinical facial eczema is still widespread from the top of the South Island to Northland. Thank you for listening to that. So now we can go into questions. We did have a few from people who, as you registered, you could ask a question. So maybe we'll go to those ones first, and then we'll go to any that have come through on the Q&A or the chat. So while I've got them up here. So the first one is, can sheep be naturally resistant to FE, or is FE tolerance limited to those flocks that have been targeting FE tolerance for decades? So I don't know if you wanted to comment on that one first, Ginny.
Yeah, and anybody else is welcome to chip in. I guess there's some argument that generations of sheep that have been raised in a high FE environment, there is going to be some natural resilience to it or resistance to it. But in terms of making really speedy genetic progress to a point where you don't see the clinical disease and the subclinical effects don't affect performance very much from year to year, there's nothing like that actual facial eczema selection program using sporidesmin and then using those high merit animals widely in a flock. So you can get a wee way, but it won't be as good. That's my answer. But you might want to add something more to that.
I am good unless anyone else wanted to. There might be some more questions around genetics later. And in terms of performance loss for subclinical FE, so this is definitely something we're wanting to kind of reconfirm in that more in-depth study. But yeah, we have looked into the existing literature as well. There has been work done on this in the past, which shows that, yeah, lambs and calves can have 20 to 30% reduction in average daily gain if they are affected, especially, yeah, with that subclinical affecting the liver like that. They're just not going to function as optimally. Dairy cows, the milk production loss can be quite significant, so 5 to 15% per lactation. And if you are a dairy sheep farmer as well, that will probably be quite similar. And yeah, don't know if you wanted to comment on those ones, Ginny.
Yeah, probably just adding to that, the variability that we see in years between ewe scanning performance. Often that's a nutritional thing too, but when it's got eczema challenge overlaid on top of it, definitely you may see very few clinical cases, but it can really be knocking your ewe repro around as well. So yeah, it hits in all directions.
Yeah, and I guess the other thing is wool. If you've got sheep with wool on, it can reduce by 10 to 20% in weight. And also having breaks in the fleece is quite common if you've got affected sheep as well. Right, so this was a question from someone who is in the study. So they didn't get facial eczema spores in their poo samples, but they still had some elevated GGTs. So is this method accurate? It's a really great question, and definitely, this is part of something we're trying to understand as well in terms of how these methods work, what is the best way to use them. For this question, what I'd be asking first is, were those GGT samples taken from the same mob of sheep that were being, the poo was being collected from? Because the mob that we are collecting the poo from, we're only checking those paddocks essentially that they're going around in. So if the GGTs were taken from animals that were from a place of the farm that they weren't grazing the same area, then the risk might be quite different. The other question would be, because GGT is not completely specific to facial eczema, there are other things that can raise the GGT levels. I'd be wanting to know what kind of level of GGT they were. So what we've seen in the study and what we've been finding is that liver fluke can raise the GGT to kind of 300, 400 in that range. So while facial eczema, you might have GGTs much higher, liver fluke is kind of that lower range, but it's still quite elevated. And there are other plants that can affect the GGT as well. So yeah, I guess I'd kind of been needing more information to understand that one completely, to say if this method was not accurate because of those differences. And Ginny, please jump in if there's anything else you want to add or clarify.
No, I thought you did a great job. I was worried you were going to ask me that question. You covered it beautifully. The only thing I would say though is please, if that happens again, tell someone so we can look at it at the time. Because one of the liver fluke cases that I was involved in helping with, that was really interesting working that out. And it was neat to be able to provide that farmer with the information. So yeah, please tell us.
Yeah, exactly. We're still learning a lot through this. So yeah, all
because I
information is
guess
great.
that's something about field studies is when we're getting data and you get so much data that we receive, not we, because I don't do any of it, I'm just here talking, but the people receiving the data, you get so much data that you might not necessarily see something like that. Whereas you on your farm, that's immediately obvious to you. So please don't take it personally that nobody picked that, but we would love to know.
Definitely. Right, and I think this next one I'm going to hand to you, Ginny. What effect does a hemolyzed blood sample have on the interpretation of FE results?
Yes, I can't explain the biochemistry behind that, but I remember when I was in practice that Neville Amies, who did the RamGuard program, would get very grumpy with us if we let the tubes lie on their side immediately. That can hemolyze. Sorry, I should explain what that means. That means when we've collected the blood sample, as it sits in the tube, all the blood cells go to the bottom, so you get this big red plug in the bottom, and then the serum sits on top, and that's a nice straw colour. When it gets hemolyzed, the blood cells have been knocked around or messed around so that some of the blood cells pop, and then the serum becomes like an orangey colour. So the way to look after our blood samples is to keep them upright for as long as possible, at least until that good clot has formed. So if we're collecting blood samples and just chucking them into a bucket or whatever, and then they rattle round in the back of the car on the way home and stuff, that's a really good way to hemolyze them. So you want to be quite particular about having them standing upright for as long as possible. I used to bring mine back and put them in the fridge for the night standing upright and then send them in the morning, and that was a good way to keep Neville happy. So I hope that helps.
Thanks, Ginny. So I'll stop sharing this now, and I'll pass over to Sonia for any other questions that we've had, hopefully, through the chat.
Yep. Cool. Thank you, everybody. Yes, we have got some cool, interesting questions for you coming from the chat, and hopefully, if anyone else, just please keep adding to that. So one here from Dan is, in the national spore maps that you showed, Cara, is it showing an average of tests or the highest for each month?
This is the highest for each month. So a really important point there is that, yeah, it's not the average. So we will be working through different ways to present the data.
Next question is, would you be better to start spore counting later on and then carry on into June?
Yes, I mean, I'd love to start in October again and then go into June, but that might be pushing it with with the farmers already giving up their time. Yeah, it would be really interesting. We had a few farmers in a couple of regions continue into June this year, and it was kind of mid-June until they tailed off. So I think it would be great to see if we could extend it even just one more sampling event in June.
And I guess from an individual farm perspective too, that the answer to that probably depends on where you are and what your FE risk is like, because you wouldn't want to sort of cut off your nose to spite your face and not start counting till March and have missed a spike that had occurred in January, which is going to set up extra risk.
Thank you. So another question, what correlations are you seeing between weather patterns and facial eczema risk? And are there any projections we should be aware of for the season 3 moving into next autumn?
That is a great question. And as you can see kind of by the maps, there are differences and we assume that will be related to weather. So we're still going through that analysis of the data. That would be a great thing to see if we can actually pull that out ahead of season 3 to provide that information. So I will talk to the team at Ingenium who have been getting all the meteorological data from the last 50 years to input into this model to see if there are any insights they can share there. So thank you for that.
Next question, nice, good genetics question for you. Is individual flock observed resilience, such as where animals haven't been introduced from flocks deliberately challenged by testing, is it more of a grazing choice or is it actually due to a genetic immunity resistance to the organ damage? Did you get that?
Great question. Who would like to take that?
I'm going to stick my neck out and say we quite often talk about that when we're talking with breeders, and maybe there is some behavioural component to it, but it seems likely that it's way more than that, because those flocks that are in those really hot areas, you know, they'll have spore counts in the millions, and those ewes have to be grazing that stuff at some point, or they're not eating. So yes, there might be a bit of behavioural stuff, but I do think it's more than that. Cara, you might have some more sort of immunological or biochemical answers you can add to that.
Yeah, well, I was just going to say, I think it's more that, as you say, we've got anecdotal, kind of not data, but anecdotal evidence that maybe that's happening, but I don't think we have enough data to actually say that grazing behaviour might be affecting that. But again, if there's any other genetics people on here that would like to have any comments on that one. Let's see the full list.
No.
And I guess I might be leading us into really murky waters here, but the fact that even though the work that Ag Research have been doing to try and get an immunological marker, it hasn't given us the gold, the ticket with that. You know, they have found differences, right, between animals of different genetic status. Sorry if I've gotten murky there.
No, all good, Ginny. Thank you for that. It's a good question, that one. So Hugh has got his hand up, so I'm hoping that Hugh, you can unmic yourself and go for it.
Yeah, thank you. I have just got a couple of questions, and thanks for all the work you're doing. Can't seem to use the chat for some reason. It's not letting me type anything, but I was just, over the last 2 years, I've enjoyed being involved with the collection process and learnt a lot through it. But just wondered, one thing was that I found really useful from a collection point of view and from a farmer point of view is when we got the results back in a week or there seemed to be a time period there where we got the results back quite quickly, and then it sort of changed, and we weren't getting the results back before we were taking our next samples, and it was cool to try and get a bit more of a real-time idea of what was going on and to try and help us make some decisions. So I just wondered if it's possible to try and reduce that turnaround time at all. And then the second question was, with all the results that go in and we get the emails back, obviously telling us what they were at that time, but it's kind of hard to keep track. And I assume you guys will have them all recorded there and recorded against each farm as well, and wondered if it'd be possible to share that information so we can look back and go, oh yeah, this is our farm and these are our results for our farm over the last 2 years, and it'd be quite interesting and potentially stuff we could use on a farm level to try and help make decisions and further understand this going forward.
Yeah, thanks, Hugh. Good comments. So the first one, yeah, so the lab they're getting extra staff because it was during that period where it just seemed to be people were either sick or away, unfortunately. So they are getting extra staff to be able to get those results to you quicker this coming season. The next question, yeah, it'd be good to know if, like, do you think a graph of how the spores are looking for your property over those fortnights? So every fortnight send a graph or something?
Yeah, or even a summary at the end, just with it all there, so we can look back on the year, and that'll be cool. Like, yeah, it doesn't have to be probably every month. You probably don't have to do it, but if you've got all the data there and you can bang it into a few graphs and send it back, that'll be cool for sure. Yeah.
Brilliant. Thank you for that. It's useful information.
Yeah. No, good. I was just pretty poor at keeping track of them. Well, I guess you're busy and you're doing other things. You see the results come through and you're like, oh, sweet, it's pretty low. Or, oh, shit, it's getting up there. But yeah, to have it all sort of tracked out over the year, and it obviously not perfect and don't get every fortnight in, but it's good motivation to try a bit harder if you know that you'll miss a spot on your graph, I guess. Yeah,
cool. Ginny, did you have something you wanted to say?
Oh, yeah, just jumping back to the genetics. Catherine McRae's online. She can answer some of that probably more accurately than I did.
I'm not sure how much extra I can add, but I guess part of it would be that with all of these disease challenges, like we know there are differences between animals, particularly with facial eczema and their ability to tolerate the challenge. So even if you haven't got a history of buying from tolerant rams, there will still be differences between animals as well. When we do ram guard, we know exactly how much sporidesmin the ram has had, whereas when they're on a paddock, there will definitely be an element of, will likely be an element of grazing behaviour as well. So with natural challenge, it's a bit harder to quantify, but Warwick, managing to separate out those two is a bit harder if you haven't actually done a proper challenge with the animals. But just because they're not from a history of ram guard breeding doesn't mean that you won't have animals that are more tolerant. I hope that answered your question.
Thanks, Catherine. It's great. We can just crowdsource.
Great. So we have just one other question that's come in, and it might be thrown open to anyone who would also like to, if anyone's got any opinions on it, but it's how long until somebody can introduce a plant genetic solution? Is that on the horizon anywhere?
I think that's targeted at me. Thanks, Warwick, listening for asking the question. So basically, a plant genetic solution, and I would say would be a fair way away. A more likely solution is the use of microbial endophytes in plants. And look, there is some evidence that some of the fungi that infect, particularly ryegrass, that don't cause disease, do have a suppressive effect to some extent on Pithomyces or Pseudopithomyces. Tina Voiser, who's also on this call, might be able to add a little bit more information there. But a straight plant genetic solution, I'd say, is highly unlikely because you're actually looking at the plant producing a fungicidal product, which is not impossible, but you'd have to understand what that product is to actually start selecting for it. So it's a long shot. The best option is finding effective fungal endophytes that can infect ryegrass and suppress the Pseudomyces spores.
I can add a bit to that, and I agree with John. I think these endophytes are a really good option. One of the advantages of them is that they are readily transferred between plants. So when you develop new cultivars, you can transfer that endophyte between cultivars, and so it's a sort of a technology that can be introduced into a cultivar to provide that extra layer of protection. And while we haven't done a lot of work up till now to look at the effects of individual plant genetics on the numbers of spores in pastures, so our data have not really been set up to collect that type of information, but we have incidentally noticed some aspect of that as well. And so I think it is something I'd like to look at in the future if there was resource for that.
Awesome. Thank you. Thank you both for answering the questions. That's awesome to have you on as well. Just is there any other final questions? You can either raise your hand or unmute if you want to.
Yeah, that's the question regarding rainfall. Is there any difference in spore count between, let's say, 1 metre of rainfall to 2 metres of rainfall, or is it more to do with the rainfall moisture levels in the ground and temperature?
Yes, so rainfall can affect, so there is evidence that if there's a lot of rainfall, it will wash the spores away. But actually, Tina, you might know the exact amount of rainfall rather than a lot versus a little. Do you have data on that? Oh, you're muted.
I don't have any data, unfortunately, on rainfall and effects on spores, on how much of that you might need. We've got sort of incidental sort of observation, is that in Ruakura, when they're in the aftermath of the Gabriel cyclone, there were very few spores there afterwards. So we suspect there was a big washout. And we do know that rainfall itself does wash the sporidesmin off spores, because it seems to be extruded out of the spore and sit around the outside of it. That's probably because it's toxic to the fungus. And so it is likely rinsed off to a certain extent by rain, even though it doesn't dissolve very well in water. But I don't know how much water you need in order to get that.
Yeah, exactly. And I will just say that there, in terms of if you've had a really dry period and then you have a little bit of water, that's quite a risk in terms of the spores will, the fungus will just sporulate then. So yeah, I'll try and get some more precise, see if we can get any more precise figures and send them out.
Awesome. I think that's all for questions.
Right, and we're right on time. That's kind of perfect, isn't it? But if anyone does have any questions later, or if they think of something afterwards, just please get in touch with us, and we're always happy to have a chat about facial eczema. So thank you so much for everyone joining. We will send the link around to the recording as well. It will be publicly available, so if anyone is keen to watch, or if they know someone who wanted to come along and couldn't make it, then they can always do that. So thank you so much, and have a wonderful weekend.